Hello, in this video we will discuss about the IVF and ICSI mechanism, the in vitro fertilization and intracytoplasmic sperm injection. These both are different, and on the basis of different complication, the diagnosis can be done by this IVF or ICSI. So let's begin to understand now. So in this way, here is the female, basically female contain reproductive system, two ovaries, and the causes for the in vitro fertilization is fallopian tube damage or blockage, so the egg will not fertilize, ovulation disorder, and endometriosis and uterus fibroids and unexplained infertility also involved for this in vitro fertilization. So the problem is here, so unable to fertilize due to the egg releasing problem. Egg will not release normally. So here is the ovary, which that's contain a primary follicle will convert into the secondary follicle, and the secondary follicle, the luteinizing hormone is used for the ovulation, while the follicle stimulating hormone used for the maturation of the primary follicle into the secondary follicle. This is the hypothalamus, which that release the gonadotropin releasing hormone normally will stimulate the adenohypophysis to release follicle stimulating hormone and luteinizing hormone from the pituitary gland, and will target to the ovary to stimulate the primary follicle through follicle stimulating hormone binding to secondary follicle, and maturation lead to ovulation to corpus luteum formation here, yellowish color. So the corpus luteum is another way. We will not understand about that, but let's begin to understand the in vitro fertilization. Remember, the test tube baby is the procedure in which the normal fertilization not occur, but the gametes produce normally. So in this way, we will treat with the analogs of the gonadotropin releasing hormone, and in this way, analog will stimulate the hypothalamus, pituitary gland to produce follicle stimulating hormone and luteinizing hormone. Our production of the FSH and LH will target to the ovary to release more than one follicles into the secondary follicle and release more than one egg. So the genetic disorder is also involving for this problem. So after we can ovulation of egg artificially and retrieval of egg through a needle and inject into the tube and provide the normal environment like a fallopian tube and uterine environment, and in this way this is the egg contain polar body during the meiosis one done here, and oocyte and the cumulus cell. So in this way we will put this into the petri dish. In the normal condition the egg is present here. So after this, we will take the semen, the sperms from the male, required male. In this way, the male sperm will be poured into the petri dish, and in this way, the one will be fertilized ultimately. And after this, the fertilization, a membrane will be formed, and in this way, the fertilization process will be done. In this way, the zygote will be formed. So after the zygote formation, basically is the cleavage lead to morula, blastula and blastocyst formation. Here you can see in this diagram in this video. So here is the blastocyst formation after the morulation through cleavage formation. And in this way the blastocyst is the type of the embryo stage which can be implanted into the uterine wall. So that is why we will able to implant into the uterus for further development. That is known as the IVF, in vitro fertilization. So the external environment we will provide to fertilization and implant that embryo into the endometrium, into the uterus, will lead to further grow and to produce mature fetus into the baby. Let's begin to understand the intracytoplasmic sperm injection and what is the reason for it. So the ICSI is used due to abnormality of sperm or egg who unable to fertilize. So for the normal, for the basically teratospermia will occur, for example, will lead to change of the sperm shape as compared to the normal shape. So here we will understand some abnormal sperm shape, for example, big head and the not tail, without tail, rough head, and on the other hand the two body or two flagella, and on the other hand the short and two head and short sperm. This is the abnormal sperms. When we will take abnormal sperms and we will take egg, if the egg is normal, if for example, so the under microscopic condition we will see, and we will procedure through holding pipette and injection pipette. Injection pipette is used for the injection of sperm, and this is the meiosis 1 is done here but not meiosis 2. So this is the oocyte and this is the spermatozoon and the cumulus cells, already I have explained. So we will put in the 3 o'clock degree as compared to the polar body with respect to the polar body of the egg. So anyhow, let's begin to understand with the visual mechanism. Here we can take the sperm in the needle, and needle is very uh, small. So when we will put the one sperm, select one sperm and inject by the holding pipette, will hold in the 3 o'clock of the egg, and injection into egg will be process complete, and in this way the zygote formation will occur, and the procedure further is same like a in vitro fertilization. And in this way we can implant this blastocyst after the complete blastocyst formation in the in vitro. So this video we have discussed about the in vitro fertilization and ICSI, the intracytoplasmic sperm injection. I hope you make sense about that. If you like this video, please make sure to subscribe, like and share, and also comment below for understanding better. Thanks for watching. See you in the next video. Bye for this time.